Okay. How's everybody doing? We are getting ready to look at some samples from Sycamore Lake, and turn this off for now. From Sycamore Lake, which is a lake in Indiana, and also have some material from a field trip that we took a couple of weekends ago with my students. And so those are materials from the Chinook Lakes, which are maybe like 20 minutes from campus. And we'll just be looking at the diatoms for the most part in both of these lakes. And this first sample that we're looking at is the Sycamore sample from Sycamore Lake. It's actually a lake on the Sycamore Outdoor Center on their property. And we went looking at it last weekend to try to see, on Friday, to see if we could use it for the next Diatom conference. And it's part of campus, so it's likely that we would be able to use it, but we just need to make sure that the facilities would work for us. And what it means is that in the fall of this year, in September, near the end of September, we're having a Diatom meeting here, and it will be like 80 to 120 Diatom nerds all joined together in a conference to discuss the research and hanging out on the sort of campus facility for the outdoor center. And so I still have a bunch of planning to do. We need to figure out how to feed everybody and what to charge everybody, and I need to get the costs, the official costs from the Sycamore Outdoor Center for like what it's going to cost us. But I think that we're pretty happy with it as our selection for where the meeting will likely take place. So that's nice, and it's nice to be making some progress on it. We were supposed to host it last fall, but because of COVID, basically, we decided to just not have the meeting at all that year and wait for the following year. And somewhere around like midsummer, one of my colleagues just sort of sent me a message and was like, is there any way we could host it this summer? And I was like, no. But everybody, the North American Diatom people, are all anxiously waiting for me to give them dates and to tell them where it's going to be. And so we took some pictures, and basically we secured the location. And then we'll be, I don't know if I'll be streaming anything from it. It's a Diatom conference, so some of the stuff on there is materials people may not want to have shared. But in the event that people are interested in having some of the events, I might share them. There's an auction, and there's a bunch of events that happen. And we should have internet that's pretty decent since we'll be on campus facilities. But, it's an outdoor center, so it's not designed for high-tech streaming. But I might be able to manage it. I'll just have to see how good the connection is when I'm out there. So we'll see. But one of the things I wanted to do today was just to image some of the diatoms that are present on these samples that I collected, and in part that's because our friend Studio Cornix, who's an artist who streams on Twitch, is going to be developing and designing some of the artwork for the conference. So our friend Studio is on the West Coast, and that's their background information. And I recommend that you give Studio a follow. It's a very chill stream. She just spends most of her time either looking at stuff that is in a microscope or drawing stuff from the things that she's looked at in the microscope, and then painting them. So she does basically paints with pigments made from the algae. And I'm her moderator on Twitch, so when she streams, I'm almost always there for the whole thing. And also, I'm always interested in seeing what she's looking through, because often there's diatoms. So, hey Nid, welcome in. You can see that I've got my automatic shoutouts working, and as soon as you say something, if you're one of the streamers that I've managed to enter into the little array, it automatically shouts you out. Our friend Nid was on Twitch, I guess that was last week. They have a homemade confocal microscope that they were streaming from as they were sort of showcasing what they could do with it. And it's a pretty interesting endeavor to make your own microscope, especially one as fancy as a confocal microscope is. So you might want to give Nid a follow. I don't think anybody else is streaming from a confocal microscope, to be honest. There's very few people who also utilize scanning electron microscopes on Twitch. And that little bell you heard is because our friend Huggin came in, and they are a subscriber. And Huggin also streams on Twitch. I don't have you in my database because I didn't actually realize that you were a streamer until you rated our friend Hannah Rebecca Music. So welcome in, Huggin, and thank you for being a subscriber here to the channel. The subscribers get the automatic little bell sound that shows up. Nid says, I just got your argon laser. Oh, cool. So hopefully you'll be streaming something from the actual confocal and let us see like cool images. A confocal microscope uses lasers instead of electrons to image things, but it can produce some sort of like SEM type of images, like similar to it's a different tool, but not completely dissimilar in terms of like the kind of views you can get, except for it produces 3D models of things, whereas we are looking at sort of topography. So the normal thing that you can produce from confocal microscopes is more of a 3D model, kind of a structure of things. So we're just looking around. I think that's true. Isn't that true? It's a still array scanning. Yeah, it's array scan. It's scanning a sample. It's like a beam scanning a sample, right? Yeah. So. All right. So this diatom that we found right here is a very common diatom, but a very cool one. This is the Gumphanema acuminatum, and it may not actually be, I'm a little fuzzy on whether it's actually acuminatum, but close to that. There's some things that are very similar to it that have been described, so I can't say with total confidence that it's acuminatum, but definitely in the range of that. And it's one of the diatoms that my students always like because of the outline. It's a very curvy outline, interesting shaped diatom. So I'm going to rotate it. It needs to come back maybe 20 degrees. So maybe it's a little bit more than that, but we'll find out. Not bad. Maybe just a little bit farther. Yeah. And then, so the speed, to change the speed, basically it changes the resolution of our image as it scans across the image. The slower rate, it will make a higher resolution image. And then I'm just going to zoom in, get it in focus, back off a little, compose the image. So once I feel like it's in sufficient focus, everything here looks okay. Then I'll back off a little bit, so we can have the whole diatom in our field of view. ideally all at once, and as close as we can get without cropping any of the ends of it off. Somewhere around here. So this is a genus that's characterized by being asymmetric in one direction. So if you were going to fold it in half, this side matches with this side. So if you drew a line that ran right down the middle of it like this, it would be symmetrical across that line. And the raphe more or less follows that line, this little slit that runs down the middle of the valve. And if you tried to fold it the other way, across the middle, they wouldn't match up at all. And it's because one end of a gomphanema has a head pull and the other end has a foot pull. So this down here is the end that the diatom usually makes its stalk from. It lives on a stalk. So I'm going to slow the beam down a little farther. I think that it's probably in good focus, but I want to get the brightness and contrast set. And then, hey, Sinophyte, how are you? I also got you fixed. I think it was not reading your name correctly before. Sinophyte's another microscope streamer here on Twitch, and just started streaming, so you should consider giving them a follow as well. We would be happy if you would do that. You can see up here in the upper part of the image, this is the foot pull, and you can see these little sort of openings, like salt and pepper shaker openings at the bottom. This is where it exudes the EPS that it makes the stalk from, and then it lives on a stalked colony for part of its life. It does have the ability to crawl around, but usually once they're on the stalk, they're on the stalk for most of the time that they're around, and they replicate on the stalk, creating like many versions of themselves as clones, just in that one place, basically. And I think the image will be okay. So I'm going to just go ahead and take that picture. So if you're coming in late, this is Sycamore Lake that we're looking at. Sycamore, like the tree, is the it's the mascot, I guess you would call it. I don't know how many places have trees as a mascot, but it's the mascot for our university. Is a tree. Not like an animated tree. We have like a, what do they call it? Magical, ambiguously defined woodland creature, which everybody, I guess, thinks is probably a fox, but it's blue colored and androgynous. They named it Sam, so it could be either Sycamore Sam the dude or Sycamore Samantha, I guess. And then refused to answer any questions about whether it's Sam Mule or Samantha, which is fine. And also they refused to answer whether it was a fox or not. So it's just a woodland creature. Foxes aren't normally blue, so that's fine. But yeah, so we don't know what that thing is. Just Sycamore Sam. Kalathon, welcome in. It doesn't look a little bit like Sonic. I think you're right. It doesn't look much like a hedgehog, though. It's Sonic-like, similar to Sonic. Kalathon, I know you asked me to try to find some leeched teeth for the stream. You gotta go fast. All right, we'll see you. I mean, if you're leaving. You're doing good? You're recovering from a cold. You spent some time watching the new Batman movie. Oh, all right. Oh, it was a joke. Okay, I see. Your college was a turkey. Well, I mean, magical woodland being turkey. I guess you had something, you know. But the search for the leech teeth is going to have to continue. Got it. Because I went down there and most of the water was very low in Goose Pond and the surrounding wetlands. So I didn't see a substantial high lake level, but I did sort of step out onto some vegetation mat and sort of reached out with a stick and grabbed some goopy stuff from the lake, which is mostly algae. and I didn't find any leeches in it. But I couldn't actually get like way out into the water because I didn't bring a boat with me or anything. But I did take some samples, and none of the samples had any leeches in it yet. So I'm gonna guess I have to come back a little bit later in the summer, um, or we could order some leeches just to have around. Sycamore Lake. I guess that's a possibility. I think they do distribute leeches in a way that you could access them. Let's see. This is Sycamore. And our sample is Gomphanema acuminatum. So this is Gomphanema. Yep, I spelled everything correctly. There you can see their typical asymmetrical shape. They're in the category of asymmetrical biraphids, and this you're seeing the whole valve. So we actually have both the top valve and the bottom valve in this case, and we're only imaged on the valve face, but you can actually see the girdle bands wrapping around it, and it's up off of the surface. So there's probably another valve face on the bottom that we can't see, but because we can't get to that angle. But I'm pretty sure that it's actually the whole frustule. These were collected live. Well, they were living at the time. From Sycamore Lake, I had some sort of a bottle in my car, and I just dunked it in the water and squished some stuff from the boat ramp into the sample. And it's filled with diatoms. If I zoom out, you'll see they're everywhere, and they're mostly these colonial things. That's a Hansia amphioxus, is a typical sedimentary diatom, edaphic diatom that lives in soils. These things are all little fregillarias and related organisms, and I found this little Gomphanema in here. I've seen a Melisira earlier when I was just rolling around looking at stuff, which is probably the dominant plankton in the lake, or the associated stream in the shallow waters. And then I also saw some little round guy, which was Lindavia, maybe tripartita. I don't know. I didn't get a good look at it. I was just glancing around and saw some round stuff. So here's a Melisira. That's a whole, the whole diatom. It's actually, I think it's actually a colony. So two diatoms, one here, and then there's a girdle band wrapped around joining them, and another 1-2 here. So they look like little pills. We've seen Melisira from Pacific Plankton's marine samples before, and they have these little star-shaped spines on their valve face, which I will get into focus for us. And they're used to basically join the valve face of this one with its neighbor, and they wrap a girdle band around it, basically. So they just look like sticks, basically. A little boring, but if you look at them on the valve face, they're kind of interesting. And I don't know, probably 9 times out of 10, this is Melisira. The most common one. Vulgare. Is it Vulgare? I'm trying to think of the name of it. Oh, it'll come to me. There's a really common Melisira. Swoobu, hi, hello. What are diatoms? Diatoms are a type of algae that make a siliceous skeleton, a skeleton made out of silica. And I think that's the command. You can go to diatoms.org. I'm a scientist. I look at diatoms for a living. I mostly look at their skeletons. I'm a paleoecologist, so I look at the diatoms as they are found in lake sediments more so than the ones that are found like living. Um, so mostly I'm just looking at their skeletal components. Try to use that to figure out what species they are, and then I use that to reconstruct something about the environment. So this diatom that we're looking at here is a Melocyra. It belongs to that genus. And if you click on that link, it'll take you to pictures that'll show you basically the kind of diatom that we're looking at right now in the in the scanning electron microscope. I also sometimes stream from a light microscope and sometimes from a stereo microscope. These are instruments that I have in my lab. So I'm a paleoecologist here at Varians. That's the diatom it is. Melocyra varians. So I need to start with a V. I was fuzzy on the name. Yeah, so this is Melocyra varians. It's a common diatom that occurs in usually moderately deep water with flowing conditions or high wave energy. And yeah, so I'm just going to image it, taking some pictures for a project that I have with an artist who is going to be rendering some of these in her style, and then we're going to use them probably for a diatom conference that's occurring here. I thought this was going to be a particularly cool way to showcase the diatoms, because we'll have diatoms from the lake where the actual conference will be occurring. So a lot of times people just have some art, but it's not necessarily tied directly to the site. And I thought it would be really neat to actually have a sample that was collected like at the place where the conference was occurring on the actual art that we have on the t-shirts for the conference and mugs and things like that. So it's pretty common for each conference to have its own like sort of art, but it's pretty uncommon for people to have an actual artist. And I think it would be fun, and especially because I like to kind of combine science and art, to have an artist who's actually there. So we're probably going to, well, not probably, we're definitely going to fly Studio Cornix out to let her join the conference. And hopefully she'll even give us a little talk about a project she's been working on, where she was going from site to site where Ehrenberg collected diatoms from, I think, probably the early 1900s. And he documented all of it and drew all these really intricate diatom images from the ones that they observed. And she sort of replicated some of his journey and collected similar diatoms and then made some art pieces around them. So the outer shell is made of glass. You did some reading off of Google. Yes, the outer shell is made out of opaline silica, which is basically like glass. It's not quite glass. It's see-through material though. So and it's silica, which glass is. Glass is basically a man-made product, but it's the cell wall of the organism. So it's an algae. They're single-celled, and they can live in colonies or they can live independently, and they're microscopic. So everything that we're looking at in the scanning electron microscope, right now this image is being magnified 7,000 times, and everything that we're looking at is very, very small. And the scanning electron microscope basically allows us to blow those up to really large sizes so that we can see all of the details. If you looked at this thing in a typical light microscope, you would be able to see this even at 400 times magnification, or maybe even 200 times magnification. It would be very small, but it would just look like somebody dropped Tylenol tablets into the sample or something. They're basically little, capsule-shaped diatoms. And so a lot of the detail is lost when you do that. And scanning electron microscope allows us to see a lot more of the detail. What's different from, glass is, like I said, a man-made product. Opaline silica is silica that's got, in the chemical formula, it has, it's hydrated. so it has water mixed in with the crystal structure. And there's a bunch of different types of opal. Usually it's like opal CT, opal T, and then if you keep putting it under pressure and temperature, it converts to, something that's a little bit more like true glass. But functionally, opal and silica is basically glass. Compositionally, they're basically the same. So there's Studio Cornix right there. She made me green. Studio, we're looking at samples from Sycamore Lake, which is the lake at the site. And there you got a little bell and a shout out. Thank you for being a subscriber to our channel. Our subscribers get little bells when they talk the first time each time. Swoobu says, is it rigid? Yes. It's glass. It's like glass. It's solid. So it's the cell wall of the organism. How does it... It's fine. You can ask as many questions as you like. How does it interact with the environment? So on the diatom skeleton, there's a bunch of little holes, which we usually call processes. There's a bunch of different types of them. In the image that's below me right now, you can see a whole bunch of little holes in the diatom that's blown up right there. That's in Actinopticus, which is a marine diatom. We can see a close-up of those little holes in the image that's below me. Those are all colorized images, so they're false color. The normal images coming off the SEM don't have color because it's collected with electrons. So there's no light. There's just basically light and darkness with respect to contrast and brightness. And that's all sort of fake topography as well. Hello, Fall Machine. How are you doing? So they exchange materials like nutrients through those openings. They can basically create a diffusion gradient, and then if there's a lot of nutrients on the outside of the cell and very few nutrients on the inside of the cell, that gradient basically pushes nutrients through the cell. So that's a normal pattern for many small organisms. This is the diatom Melissyrovarians, and it's the whole cell. It's actually a small colony. It's the external view of the girdle band view of the diatom. If we looked at the valve face of this, it would be circles. So this is a prism. It's a columnar prism that's basically rounded. So the diatom is basically from here to there. They're like little pill-shaped things, and there's one here and there's one there. So two valves are present, or two frustrals are present. And then this thing that's right next to it here looks like it's maybe a navicula. I'm going to slow the beam down a little and see if I can see any of the details. I think we can. I got to refocus the image a little bit, but you can see the raphe, which is another way that they kind of communicate with the outside world. For some of the diatoms, they have an opening that runs through the central sternum on the edge. And they use this basically to crawl around. So people don't often think of algae as being able to crawl around, but diatoms can. Most algae have some sort of a flagella or ciliate-like structure on them, but diatoms lost those structures a long time ago, and they don't have them present on the valve at all. So, but you can usually tell whether a diatom is plankton or benthic based on its shape and the presence of one of those raphes. So if it has a raphe, it can crawl around. If it has those slits that run through the cell wall, they can crawl around. And if they don't have one, then they can only float. And so this little guy here, is plankton. It doesn't have any kind of a raphe. It's basically just a circular diatom. This is a Stephanodiscus, is the genus. And so that's plankton. That's a benthic diatom that lives on the bottom, and this is a partially planktic, mostly benthic diatom, but they can get detached. And then if I zoom out, you'll see that most of the slide is composed of these like chain-forming diatoms, and this was taken in shallow water. So I would expect that it would be dominated by things like these that live in kind of intermediate depths in the water column, or things that are very shallow, like the benthic diatoms. And so it's like a mix of what occurs in the lake. There's a little Cerulella. This used to be Cementopleura, but it's in the Cerulella category now, I think. Or Ichinella. I don't know where all of them went, but they've been transferred relatively recently out of Cementopleura. So probably a Cerulella. And then you can see almost all of the sample is composed of these. This is a Melisira like we were just looking at, or these Fragillaria that we can see that are basically all over the place. So these are the most common things that are present in the water. And occasionally we'll see some of these naviculas. If it didn't have some junk on one end of it, I would really like to get a picture of one of those that's nice and clean like that. So I'm kind of just scanning around, looking for things to image that are in good shape. That's a nice close-up of, that's a Gomphanema. There's a little hole here. This is like Parvulum, in the Parvulum group. And then you can see these ones have like overlapping little spines that they use like zippers. It holds one valve onto the next, so you can kind of put your knuckles together like that, and you can't pull your hands apart. That's basically what's going on with those. They have little spatulate endings, so they're held together, and they don't fall apart very easily, which is why we keep finding them in the sample in these big colonies, because they are difficult to separate. And sometimes these things can live floating in the water column. This is another Gomphanemoid. I think this is actually Gomphanella. I don't think it has... I think if we saw the valve face, I think it would actually be missing the stigma. I think that's probably Gomphanella, something in the Olivaceae group. You can see the pores on these ones are basically C shapes, little C's, that are going in every direction. So sometimes the pores are simple, sometimes they're quite complicated, but that still has a bunch of junk on it. So I don't want to image stuff that has junk on it. I want to try to find things that are nice and clean so we can see the detail. And hopefully Studio Cornix will, if she wants to include the detail, she can have it. So that's nice. So I'm just going to take a bunch of pictures from this, and then I'm going to send this off to Studio as a microscope slide, and also send her the SEM images from these. So she'll have plenty of time to stare at it and think about how she wants to compose things. Give me a second. We'll try to find us something to image, and then I can come back to chat. It's difficult for me to see what's going on when I'm simultaneously running the scanning electron microscope. It kind of takes all your effort. This is Hansia amphioxus. It's an edaphic diatom, usually found in soils. It also has junk on it, and I zoomed out, I saw something over here. This is a navicula, another long skinny navicula right here. Also has some junk on it. The samples were collected, as I mentioned, just with a bottle or a Ziploc bag, and I just stuffed some algal material from the lake into them, and then they were digested chemically using nitric acid and a hot plate in the lab, and that was done yesterday by my lab tech, Mallory, who at least once a week I throw some random samples at her and say, can you please turn these into SEM stubs for me? And magically, within a day or two, I usually have something for us to look at on the scanning electron microscope, which is nice. So I swear I saw some round things in here, and I wanted to get some images of the round things. They're just pretty uncommon because in the shallow water we mostly get these zipper colonies of things which are everywhere. I can also stop and take some pictures of those because I feel like they're the most common thing. I probably should have some images of them for studio as well. But since they're everywhere, they're easy to get pictures of, and I wanted to try to find something that was a little less common. Also in the mix, I see these long skinny things are ulnaria. Here's a fragillaria that's not attached to a colony, and there's also some little things that are hidden in here, in amongst the debris that I see occasionally. We'll zoom in and take a look at some of these zippered colonies, just so that we can get some images of them, and I can come back and see what's going on in chat without leaving you guys for too long with no one answering your questions, if there are people asking them. So there we got little zippers holding the valve faces together with spines, basically. Trying to get it to line up right in the middle is sometimes a bit of a challenge. Okay, now I'm going to just zoom in. We'll do a focus. Somewhere in there. I'll zoom back out. I'm going to try to adjust the brightness and contrast a little for these. They're just a little bit dark, a little on the dark end. Sometimes when you change height dramatically from one thing to the next, the brightness and the intensity need to sort of be adjusted again. So I usually just push a button and let it do that for me. Sarah, hello. Sorry, I didn't see you there. I'm coming. Give me a second. This looks like it's going to be in focus and ready and good. So I'll just go ahead and click that. How are you doing? Let's see. How do they grow themselves to be so symmetric? If it's rigid, how do they grow at all? Do they grow like snowflakes? So to answer your question to the best of my ability, Swobu, they divide by creating a new valve inside of their existing valve. So their valve is shaped like a Pokeball or a hat box or whatever. And so it has an opening in the middle part, which is where the organism lives. And when it goes to divide, it basically starts to pull itself apart. And then it will create a new valve facing the old valve, internally on one side. It's hard to show. And then on the other valve, the same thing will happen. It will create a valve inside of the other one. And then both the valves will pop apart, and they actually shrink as they get older. They don't grow because they have a rigid skeleton, and growth isn't possible, as you've recognized. So instead of growing, they shrink. And the population gets smaller and smaller and smaller, and then eventually they reach some sort of a size threshold, and they stop cloning themselves and they switch to sexual reproduction. And when that happens, one of the frustules will become male. It will split open, gametes will come out, and then they'll find female frustules to impregnate. And basically, if they're planktic diatoms, this is the pattern, then they will create an egg that will grow to the maximum size of the diatom, and it's not silicified until it reaches its maximum size, and then it silicifies the outside of it, and then basically it starts the process over again. And there can be many, many generations of division before they get back to growing, and sometimes it takes them years to get to a sexual regrowth, regeneration for their size. So they don't grow bigger, they just grow smaller. Lots of green algae have flagella, yeah. Random question, has there been any diatoms that evolved into symbiotes? Yes. Well, there are both diatoms that have been that have a symbiotic relationship where they've been captured by things, and there are diatoms that have endosymbiotic relationships with cyanobacteria. So the smaller things have been incorporated into them, and then there are some sort of naked diatoms basically that have been incorporated into, I want to say foraminifera. I can't remember exactly. It's something in the marine realm, but they do have some sort of endosymbiotic relationships with some other organisms. Um, on the on the organisms that they capture, they're nitrogen-fixing cyanobacteria, so that they can get access to the nitrates that are produced when they fixate nitrogen from a gas state into a nutrient. So that has to happen in anoxic conditions, and inside the diatom, probably they generate some place for that cyanobacteria um, to function, and then they capture them and basically integrate them into their life. So the cyanobacteria have a home, safe place to live, and they probably pump some sugars into them, and then the cyanobacteria then will generate nitrogen from nitrogen gas, which most organisms cannot do, but cyanobacteria can, as I'm sure you know. Yeah, yep. So the ultrastructure is fascinating. Hi, Lisa. How are you doing? How do you age the samples? How long can a frustule last on average before it begins deteriorating? So when we analyze samples and we try to produce an age model, we do that from the surrounding material. So we don't actually date diatoms, but we date things that are found in the same sample with the diatom. So typically, like a lake, if I were to collect a sample around here, I would either analyze the carbon mass from like a piece of plant, a leaf, a twig, charcoal, something that's basically trapped in the same horizon as the diatoms are, and then we sample several discrete layers, and then each one of those gets dated, and then hopefully they're in chronological order, and we use that to create a model by approximating what happens between them. Today they use this very clever Bayesian technique where they put all of the possible explanations in, and then it gets rid of ones that basically have temporal inversions, and it results in the most parsimonious agreement between each sample. Sometimes there'll be ones that are from like old material that was brought into the lake that was laying around in the landscape, and so they'll have an erroneously old age. But generally speaking, then we use that to create a model for every sample. And the more samples you have ages for, the more high resolution you could potentially reconstruct the dates for. So that's if you're using radiocarbon dating. You can also use things like lead-210 dating, in which case we're looking at the amount of lead-208, which is a byproduct of the uranium decay series, and the transition from radon, which is a gas, into lead-208, which is a solid, means that it's constantly raining out of the atmosphere, and it rains into lake sediments, and it creates basically amount of background material. So the transition is actually from radon to, I want to say radium, which then decays to, maybe I got that backwards, but then it eventually decays into lead-208. It takes about 22 years for the half-life for it to decay to a stable condition, and then it's 210. Turns into lead-210, and lead-210 basically goes to 208. And we can look at the relationship between 210 and 208 to get an estimate of the age. Usually that only gives us about a 200-year window, and then so anything that's 200 years old or less, we can use lead-210. If it spans back to 55 to 60,000 years, you can use radiocarbon dating, so you're getting some pretty old stuff by that point. If it's longer than that, we usually have to use something like OSL, a different part of the uranium decay series, or beryllium-10. There's a bunch of other things. It's actually kind of a challenge to date things between around 50,000 years and kind of around 1 million years. And then uh, there's there's tools you can use, but it's harder to find materials that work perfectly in those sort of time ranges. And then once you kind of get into the older, really old stuff, there's some other tools that are typically a little bit more accessible. But it's put together basically by combining all of these things into a model for what we have as an understanding of how it works. So this is, I'm going to just call this Fragillaria, but I don't know for sure if it's Fragillaria, because I can't see the valve face, and it might be something closely related, but this is a colony. It very well could be something like Brevistriata, which is a pseudostar Syrah, for example, but I'm only really good at this when I can see the valve face. From the side, it could be anything. It's more challenging, basically. Let's see. I think that's how long do frustules last on average before deteriorating? So if you were to look at an individual out in nature and collect it, and you're trying to say like how long before it, an organism won't be able to live any longer or whatever, or it starts to break down, the silica stays around forever. So you can find 10 million year old diatom skeletons. It's not going to deteriorate unless you put it around something that basically will dissolve silica. And it's a little bit harder to find things that dissolve silica, so they usually preserve. In basic conditions, like acid-basic conditions, they can rapidly decay, and the skeletons will disappear pretty quickly in those conditions because it dissolves silica. But generally speaking, the process for like diatom skeletons sticking around on the surface is, there's some that are dinosaur age, like late Cretaceous age skeletons that are perfectly or nearly perfectly preserved. In terrestrial settings, I think the oldest known deposits, if I remember correctly, is 82 million years, which is there's some deposits from the Wombat Formation, which is one that Pete Seaver looked at, that are in that range for terrestrial deposits. I think that's the oldest. If you go to the marine realm, it's much older, like Cretaceous, dinosaur age sort of stuff. So substantially older stuff can be found in the marine realm. Largely, we believe that's because diatoms have an origin from that is marine, like they started off as marine organisms that moved into the land. I'm gonna rotate this ulnaria around and try to get a picture of it. I want it to be maybe 30 more degrees. It's not terrible. And then I just need to zoom in. I'm going to get it into focus for us. You can see some of the structure for the striae here. I think that's probably as good as it's going to get. When I zoom out, the focus won't matter quite as much, so it'll be almost all in focus anyway. This is the internal view of an ulnaria. This long skinny thing is ulnaria. These things are little fragilarioids of some type. And then I think that's a Nitzia, but I'll need to see a little bit more closely to make sure. This should be good. Just grab a photo and come back. Sarah wants to know, is it possible to turn dried blood smear slides into SEM samples? Yes, we've looked at blood before. Hey Freeman. Um, Mallory donated some blood. She like pricked her finger and put it on a stub and dried it. Um, the problem is that it just looks like blobs, and when you put blood in a vacuum, the platelets explode or implode, right? So um, it doesn't end up looking very good. Uh, is there sexual selection in that process, or is it more just sexual recombination? Um, well, I guess there's the ability for them to find a mate, so there's some selection. I don't know that they actually seek out like the strongest diatoms and try to mate with them or something like that. But there's some advantage to being a gamete that's more functional than other ones, I guess. Um, I mostly focus on the other end of things, Freeman. I mostly focus on the skeletal material, so I don't know that much about it. Okay, you still don't understand the symmetric nature of the patterns. Well, that's just the way they're formed. Humans are symmetric. Well, like most things that we look at in nature are actually symmetric. Humans have bilateral symmetry, like, right? You can divide me basically down the middle, and I'm symmetrical. It's the nature of most organisms to have some symmetry. Things are usually in nature either by bilaterally symmetrical, or they have a single plane that you can divide them down, or they're radially symmetrical, like starfish are an example of that. They have radial symmetry, but most things are symmetric. Diatoms just actually have both types. They have symmetrical, like a down the primary axis type of symmetry, and they also have some that are radially symmetric. So it's just the way that they're shaped. I don't know. There's nothing unusual about symmetry, I don't think, for organisms. Um, so yeah. So Lisa asks, how accurate can you get with those two kinds of dating? Um, Radiocarbon dating usually has an accuracy depending on where you are in the calibration curve, because it's pretty complicated, but to answer your question as best as I can, the production of radiocarbon in the atmosphere changes through time. And so we can't use it as a perfect tool. In times in the past when productivity was higher or lower, it causes the age to sort of fluctuate a little bit in terms of like how we can interpret it. So there's times in the past where over that 50,000 year period where we can be very precise, and probably the error is something like, like calendar years, something like 30, plus or minus 30 years would be fine. And there's times where it might be plus or minus like 150 years. So it can be variable depending on where you are in the history of it. The way that we know that information is there are things that have annual growth bands, like trees, and you can chop a tree down and you can count back how many years the tree has lived based on the growth bands. And we have trees that have lived thousands of years. I think bristlecone pine has an age of something like 10,000 years, the oldest trees. And so it has these massive tree rings, and you have something that's made out of carbon, and it's in the age of radiocarbon. So you can actually take radiocarbon samples all the way along the stump of the tree and match the age with the rings that have been counted. And you can do that with many, many trees. And you can actually come up with a calibration tool for using radiocarbon to be a direct link to the calendar year. And that's basically how they do that. It's a that's how we gauge accuracy. So if you're wondering, like, how do we know that that makes any sense? We have a whole bunch of tools that are like this, that have incremental growth. So there's incremental growth in corals, there's incremental growth in trees. Also, lake records can have annual banding in them called VARBs, And people use those in places to actually count back, and then they take a sample from that interval, um, with the number of annual growth bands, or in this case, the lake deposit bands. And you can take a sample from there where a piece of wood has fallen into the lake, and you can count back the number of bands and then do the radiocarbon analysis to see how accurate it is. And it's a way for us to basically combine tools to determine accuracy, because you need independency. You need to have some independent way of determining the age of something. So, the accuracy for lead-210 is usually plus or minus 2 years near the top of the core, and maybe as much as 10 or 15 years off by the time you get to the bottom. But you can use some other tools to date that as well. They use cesium-137, which is related to when nuclear bomb testing happened, and they can actually see spikes from nuclear bomb testing that show up in lake sediments because that had a global impact. And so they can use that to produce, like if you're looking at the past 200 years and you want to know like when's 1945 or 1967, you can get a pretty good accurate estimate of those things because we have evidence of them occurring. From when people were actually, on the landscape doing things that modified the landscape. So cesium-137 is a tool people use in combination with lead-210 to produce a pretty accurate estimate of the age of sediments for the last 200 years. And then for radiocarbon, they usually use some other, like I said, incremental growth tool, or they will do 2 or 3 different types of dating to determine the accuracy of things that may be a little bit less reliable. But to answer your question specifically, it's usually pretty good. Sometimes the ages may be off if there's what we call a reservoir effect, which is basically there's a bunch of old carbon on the landscape which dilutes the amount of carbon in the sample that should be radioactive, and it creates a bit of error associated with the ages that we get. But usually we can account for that, so you can tell that the ages are off, because things that we know the age for, like the surface where the core was collected, should have a specific age. And if the date you're getting back says it's 200 years old, but you know that it's only like 10 years old, you know that there's some sort of offset with the total amount of old carbon on the landscape that's creating a problem for you. So there's ways that we can basically analyze it and determine the accuracy and use that to basically improve, or at least have a good understanding of where our potential error is in the age models that we have. So hopefully that answered that question. That's a nice internal view of a navicula. And some examples of some of these. This is the thing that we're seeing in stacks. This is an individual one of it, I think. Trying to figure out if that's actually what we're looking at. I mean, it looks like it. It's just a fragile area. So, likely, that's what it is. But I think these are the things that we're seeing. Like, this is an example of one of these. There's also some things in here that aren't diatoms. That's a chrysophyte stomatocyst right here. So the little opening, like a little Christmas ornament, basically, shape, but then it has a little opening here, which is where the organism climbs into and out of this little submarine tank that it climbs into to overwinter, usually in environments where conditions aren't good for them. Like either they like it to be warmer or something like that. So they'll occasionally make these little cysts, climb in them and hide in them. I'm not sure what this thing is, so I was just zooming in to take a look at it. It's something very lightly silicified. It looks like maybe it's a Nitzia. Just gonna zoom around a little bit more, see what I can find, and hopefully we can find something to get some more images of. And then I have some other samples that are in here, as I mentioned, so these are all from Sycamore Lake, which is on university property. So I could collect it without any concern. And we're potentially working to try to, I don't know, utilize the Sycamore Outdoor Center a little bit more for my classes and things like that. So I'm trying to develop a little bit of a good working relationship with the people who run it, so that we can potentially utilize it for some of my classes that have lake or water related research. And then rather than going to some random place and looking at stuff, we'll actually have a consistent place we can kind of regularly return to. And I think they would appreciate having people who were regularly using it. So that's sort of my plan. This is the navicula. It's the internal view. All right, let's zoom out a bit. One thing I didn't notice is any like super large things except for those ulnaria. Like, I think we saw some Cerarella up here, but not a lot of them. You can see in the middle of the slide there's also like a lot of stuff going on. It's a little too dense. So I'm trying to sort of stick to the outside edges where the concentration of diatoms is not quite so high, so that I don't have like gazillion things going on. Actually, maybe that's not a bad idea, though, is to do a picture sort of in the middle of all of it. Zoomed out a little bit, so we can kind of just sort of see the landscape a little bit. Let's do that. Might actually be kind of interesting to see it that way. So we'll do one of these. An overview. Okay, I'm going to try to catch up with chat. I know I'm super far behind. Can you show us an example of how removing erroneous information works on a small sample? Do you mean for dating? I could try to find you an example of it, but type of Bayesian reasoning that eliminates erroneous stuff. Yeah, so we use a tool called Bacon. It's made in the programming language R, so I think it's just spelled like it sounds, Bacon. Blood is boring, yeah. And that's the tool that does this sort of Bayesian technique. I have a slide somewhere that showcases how it's done. I'm not sure I have it in my actual hard drive. Give me a second. I'm never going to catch up with chat at this rate, by the way. Oh, there's no PowerPoint on this computer, so I can't really show it. I'll post it in the Discord if you want. If you want to drop by the Discord, Freeman, I'll post it up there. It's a nice little graphic that kind of showcases what it does, and I'll also send you a link for Bacon in R. There's another program as well called Bcron that does a similar thing. Both of those are pretty common. So, Sarah asks, if I have slides of lizard blood from years ago, it wouldn't look too interesting. Probably not. I don't know. And we can always throw it on here and see. What kind of ecological questions are you and your lab researching, looking at old sediments? Um, my lab is looking at a whole bunch of a variety of different environmental information. Mostly we're trying to reconstruct things like drought history, water quality. I have a proposal into NSF right now where we're trying to reconstruct the whole food web for Lake Tanganyika. So we're actually trying to use all of our information that we have, some DNA information, a diatom skeleton, some nutrient information, to basically reconstruct the entire food web through time. But more typically, we're trying to reconstruct the climate for an area that's related to, history. So drought history of the Rocky Mountains, drought history and water history, hydroclimate of the Sierra Nevadas. We're looking at East Africa with a bunch of stuff in the East African Rift that I specialize in, so there's a ton. Hey, GeoJim, hello. Thank you for dropping by. If you're a fan of geology and you like to check out really cool streams, you should visit our friend GeoJim. Give them a follow, and then when they stream, you'll be able to see some really cool stuff. They're at George Mason University. They are sort of like the coordinator for some of the labs there, and they do really cool scientific outreach. So please, if you're interested in science streams, give them a follow. Lisa, I did an insect residency with them. Oh, that's cool. Well, that's cool. You and Studio kind of know each other. Yeah. I'll work on it, Sarah. I'll post it into the Discord, but it's a pretty simple tool. So, yeah. You guys know each other. Cool. Oh, am I caught up? I feel like I'm caught up. That's nice. I'm like constantly trying to get to the bottom of talk about rocks and stuff. Yeah, of course. The geo part is obviously for geology, for GeoGym, and I'm also a geologist by training. So it's nice to hang out in there and just sort of see what they're up to. I think they usually stream on Fridays. That's when I see them hanging around. But if you have other times you wanted to tell people about Jim, go ahead. Feel free to advertise for yourself as much as you need to. This is an overview. I don't know if there's anything in particular that you want me to try to image here, Studio. I'm going to just look around a little bit more and see if I can find some of the little round guys that I've been looking for, and see if I can grab you an image of one of those, just to have something that's a little bit different content from, we have a Gomphanema, we've got some Fragillaria, we've got the Melisira. It would be nice to also have something here from the centric stuff that's not a shallow water centric or flowing water centric. See if I can find one. Maybe a bit of a challenge. So now that I've caught up to chat, I can now once again get really behind in chat. That's my plan anyway, apparently. Oh, here's one. It's Lindavia ocellata. Yeah. So got a little bit of junk on it, but we'll take it. So I turned the beam intensity up so I can kind of look around a little bit. It draws a bit more clearly when I have the beam intensity up. Once I zoom in, I actually kind of need to decrease the beam intensity so that the resolution increases when I slow the beam down. So I can get it in a nice clean focus. So the little box that I pop open is just helping me. It only redraws what's inside the box when it scans. And then I can slow the beam down and make sure that everything is more or less cleanly in focus inside the box. And then once I'm happy with the focus, I usually just let it do the whole stage that way. And beam intensity is lowered, but you can see that there's some bright areas on the outside of this that I don't want. So I'm just going to do a really quick brightness and contrast adjustment, and that'll get us a little bit closer to where we want to be. And then I can slow the beam down. So we can see the details of the valve face of the thing that we're looking at, and I'll probably just do another brightness and contrast setting really quickly, because this one also has a little bit of adjustments that it made for the the faster speed. And then this looks like it worked. I feel like we're going to have a good clean image. Up here drawing so I can take a look at what it'll look like. And speed set, light set, go. This is Lindavia, a diatom genus that's defined by the position of the rim of Portula on the inside of the valve, but I already know which species it is. So, but the genus is Lindavia. This one. And you can see some examples of it in the image below me there from the web page, or you can click on that link if you'd like. I know you love a good centric diatom. So here's a nice clean close-up of this Lindavia, this ocellata, and this is an awesome way to spend your lunch. Oh, cool. Excellent. Thanks for hanging out with us, Lisa. Think you should give the diatom... Oh, yeah, that would be great. I bet they would love it if we just took one of your prints and poster size, blew it up, and they could put it up on the wall there inside the actual little lodge. But anything that we give them probably would be fine. They'd be happy with it. Tuesdays and Fridays at 3 p.m. Thanks, GeoJim. A good variety is key, yeah. Burn in mad trees. Yes. Yes, you're very funny for a 3-year-old, and you lasted for a couple of seconds before you got banned, so congratulations. I guess that's their challenge in life, is living with himself, you know. Must be very difficult. They have medication for Tourette's, I think, if it's a medical condition. The I haven't seen any Cimbella studio in the sample at all, so I'll look around a little bit more, but I saw mostly, the only thing I have an image that I saw pretty commonly was just some plain Navicula, so. I mean, we have Cimbella here. I have some samples with Cimbella in them. They aren't from this site, though. It's also partly how I collected it. So the sample was collected from a boat ramp and like floating algae. So I would expect to see sort of a different kind of group of things, more in the metaphyton range, that would be present here. But I did get some Gomphanema and Melisira, a bunch of Fragillaria. This is Lindavia ocellata. And there are some Navicula in here, so let me see if I can turn up the beam intensity. We'll see if we can find one that doesn't look too bad. Also thought I saw some Alicocera, but maybe they were just Melisira. This is another type of Gomphanema. This is Truncatulum. I haven't seen very many of these in the sample, but we'll take a close look at it. You can see little C-shaped. Perfect. Some little C-shaped areolae on it. It's got a little bit of junk on it, but I think it'll be okay. So Gomphanema characterized by an asymmetric form. They have a foot pole and a head pole that's very distinct, and then a dividing line that runs down the middle, the raphe. And you can see these ones have these little C-shaped areolae on them that can be grouped together in rows, lines of openings on the valve face, which are the areolae. Oh, it's a sine and cosine joke. I got it. Thanks for that, Robert. This isn't really a math channel, but we'll take it. This is you get studio started, and she's gonna start telling us some dad jokes. So be careful. I like that you prefaced it with, "This is a joke," just so that we all knew. You have a work meeting, so I'm protected from your jokes today, I see. Dodo Ob Ob. I tried to find Sycamore Lake with an internet search, but I did not find any. I'll find it for you. Hang on. It's in Brazil. Sycamore Outdoor Center is the name of the place that you need to Google. And as I mentioned, it's part of the Indiana State University campus. That link didn't work perfectly, but if you just copy and paste it, it'll get you to a map of the Sycamore Outdoor Center, and the largest, it sort of has like a fork top bird foot shape to it, is the lake that is Sycamore Lake, and then there's a bunch of little lakes around it. It might not even be labeled on a map because it's such a small little system in here, but most commonly these lakes are old coal mine pits, strip pits that people basically just filled in with water. So they have a weird shape, probably because there was a coal seam or something that they were following. So did I study biology? No, not really. I'm a professor. I mean, my specialization as an undergraduate was geology, and for my master's was geology, and for my PhD it was paleoecology. So, there's a connection between ecology, biology, and geology that I sort of fit in between. So I'm more of a paleontologist than a biologist, if you're looking for like a field of study. But I need to have some understanding of biology and ecology to be a paleontologist, right? Because there's organisms that I'm studying, but I'm looking at their history, right? So I need to be able to link ecology with past climate and long-term understandings. Yeah. Thanks for that science emotes, fall machine. Um, None of the sciences actually occur in a vacuum. They all have some overlap, but I feel like the geology overlap one has the most with everything else because it's basically just a study of the Earth. And so there's chemical ways to study the Earth, and there's biological ways to study the Earth, and there's just rocks, basically, which are just pure geology, like mineralogic ways of studying the Earth. And all of those things kind of come into play with water environments where I generally work. So this is Gompphonema. I misspelled that. Hang on. Truncatulum. Something like that. Truncatum or Truncatulum? Anyway, it's a Gompphonema. If you know the answer, is there any chance to tell you what's the difference between RNA and DNA? Okay, well, that's something you could Google. Um, but, you know, generally speaking, DNA is, unless this is a joke. Are you trying to trick me? Is this a joke? Because you did tell a joke earlier. The DNA is the, you know, genetic information that organisms are made up by, and RNA is like how it gets replicated, basically, messenger information. So I can't answer your question in a more detailed way than that. But if you're telling a joke, though, it's fine. Oh, no jokes, okay. Hey, Boba, how's it going? Look at, they got an instant shout out from my my shout out code. Yeah, so DNA is the stuff that makes up, you know, like what you are, and RNA is how the DNA gets copied, replicated, basically. So it's another aspect. It's inside the cells for organisms, but really that's more of a biological question. So I would leave that to some biologists to tell you the real differences between those aspects. What's this? Hello. Hello, little Stephanodiscus. We saw a Stephanodiscus earlier, but it wasn't nice like this one. This is a nice one. Ready for imaging. It's got a little bit of junk on it, but you can see the valve face very clearly for most of the diatoms, so we can image that. It's going to be a good little image, something different to include in the sense. Boba, how's it going? Are you streaming later today? I forget what happens when people stream, but Boba does drawing and painting on Twitch, and he sort of does this outlining thing, transfers them onto laminate by carving them, and then uses that as a template to paint through over top of some abstract sort of backgrounds. But it's very interesting to watch him do it, and I hear the conversation in his channels is also spectacular, so you should give him a follow. You're currently drawing a gnat catcher to cut onto a stencil later. Perfect. I have some pictures of gnat catchers. Some from the Southwest that we imaged, and it's pretty common there. And I think I might have had a gnat catcher or a fly catcher from my yard before, but all the fly catchers are like, I mean, it's like dusky fly catcher and smoky fly catcher. There's like a gazillion of them that look exactly the same, and I think they're mostly told apart by the way that they do their like calls. So, oh, a blue-gray gnat catcher. I think I actually might have a picture of one of those, but they're really tiny birds anyway. I mean, if you eat gnats for a living, you probably don't get very big. I think I might have a blue-gray in my life list of things that I've imaged. I'd have to go looking. When we were in the Southwest, I sold so many different birds that I'd never seen before. And so I just, was just going through a giant checklist, marking off stuff I'd like one after another. I saw some really cool stuff, though. I was on sabbatical in Arizona for 4 or 5 months, basically. So I saw some really cool stuff. For people who aren't familiar with me, birding is sort of a hobby. So I do a bunch of photography, post all that stuff to Instagram, which I'm sure the little link for Instagram has rolled past us a bunch of times in the chat. I have an Instagram for the SEM account, which is basically the pictures that are below us, which I periodically will update here. And then my Instagram, which is mostly my photography for birds and moon and whatever I feel like taking a picture of, macro photography stuff usually. So you can feel free, if you're interested in photography, take a look at that stuff. I'm told that the images are pretty good, but I just take pictures of things because I like it. So Stephanodiscus is the genus, and I don't know, it looks sort of like Medius or something like that. I don't know what the species is. It's nice of it to be completely exposed and not covered with junk, or not completely covered with junk anyway. Oh, this is like a planethidium. So the valve face is multiseriate. The striae are multiseriate, and it has like sort of a area here which we would usually just call hyaline, which means there's nothing on it. Let's see if I rotate it like this. I rotated it out of the field of view. Uno momento. You can sort of see. Oh, it'll be better when I get it. So you can see the hyaline area right here. There's nothing on it, and it's basically inside. There's a structure called a cavum, just basically a horseshoe-shaped opening, like a cave, which is where it gets that name from. But we're looking at the outside, so you can't really see that structure. But in the light microscope, you see through them. It would see like a U-shaped structure, a horseshoe-shaped structure right here. And I could figure out that it was planethidium because they usually have multiseriate striae, and they're small. Like this one's pretty small, and bilaterally symmetrical, so the outline could be divided this way or this way and still produce the same type of image. Across the mirror plane, basically. Hey, Michael Learning. Look at that. You got automatically shouted out by my automatic out-shouter. Give me 1 second and I'll come back to see what you're talking about. I'm trying to get this to image the surface of this planethidium. I think that's going to be fine. Speed 7. What's the main building material these creatures use? So they have a silica cell wall. So you've been lurking and cooking. What are you cooking for us, Micah? They have a silica cell wall. So all the things that we're looking at are silicon dioxide, basically, or opaline silica. So they, like clams and snails make their skeletons out of aragonite or calcite. Diatoms make their skeletons out of silica, for the most part. The shoutouts are working wonderfully. Thank you for helping him set them up. Looking forward to testing them. It's nice when it just sort of works, right? I don't have to shout people out. My moderators can just fall asleep and then, occasionally smash bots or whatever if they have to. Since Sycamore Lake is artificial, were the diatoms already in the underground and revived to start growing again, or did they blow in from somewhere else? This is a really good question, Dodo Ob. The the water that fills the pits is probably from rainwater, but they might have brought some water in from outside and started the process. But they weren't trapped in the sediments that were already there. So the diatoms basically will colonize a water body pretty quickly. And your question about whether they were blown in, like how do we get things living in locations where they didn't start out there, is tied directly to how we interact with that water environment. So birds can carry stuff in on their feet, like they wander around in one lake system and then they wander around in another lake system, or on their wings or whatever. Fish will have diatoms partially digested in their guts. Some of the diatoms can actually pass all the way through the gut of a fish and come out the other end and be okay. So if they stock the pond with fish, that will bring some diatoms naturally into the system through the fish guts. And then some of them probably do get just blown in and will colonize the environment because they're dust-sized particles. They can be blown in by the wind. So, and also people can bring diatoms into the environment. So if they mucked around in another lake, or they brought a boat from another lake over to anything where there's material that gets transferred from one environment to another, has the ability to also transfer species that are dust-sized from one environment to another. And diatoms are underrated with respect to being extremophiles. They can handle living in some really extreme conditions. And one of the things they're really good at is coming back from being dust, from being blown around in the atmosphere. They can basically, they have evidence of diatoms that, insisted, and then they took them out of herbarium collections that were, they were being stored in baggies and books that were hundreds of years old. And they add them back into a fish tank with some water and some nutrients and some sunlight, and the diatoms will just start growing again. So, and they have a whole field in ecology called resurrection ecology, talking about organisms that have this ability to basically turn themselves off, make themselves encapsulated so that they can be preserved, and then when they get into some environment that's functional again, they can actually come back to life. So it's actually a very interesting question. But you could take a bucket of water and fill it with, just rainwater, and leave it out on your backyard and come back to it. In a few months, it will have diatoms growing in it. A bunch of other things too, not just diatoms, but all kinds of algae growing in it. And it's just because, people, organisms, whatever, have visited it and transferred stuff in, and some of it actually just gets blown in from the wind and survives. So it doesn't come from the actual old sediments, though. The sediments that were there in the lake, if they were looking at coal and it's in Indiana, those are probably Mississippian or Pennsylvanian in age. So they would be too old for terrestrial diatoms to be living in the sediments. And additionally, I don't think you'd be able to bring them back to life from that sort of a time frame. The biological stuff is probably very gone by that point. Over millions of years, it's questionable whether any of the DNA would even be preserved in those environments. So most likely those are things that were transferred in by people or birds, or anything that was potentially anything that's crossed through the environment. But most commonly we try to blame people, birds, and fish. Those are the most likely things, besides wind, that are likely to transfer something into a lake system from you know, from wherever. And I will tell you that I have a fish tank at home, and if I have it in the sun, diatoms will grow on the sides of the fish tank. So I didn't put any diatoms in there. We just had fish that were in there and fish food. So I think that same thing happened in my koi pond. We just got diatoms growing in there, and I have no idea how they got there. It started off as like a, you know, piece of plastic on the bottom of my yard. They definitely weren't living in the plastic. So it's a really kind of interesting question about how do organisms disperse in environments, and how do they get into and colonize environments. But diatoms are actually really, really good at colonizing environments. So they can, if they can find an environment that they're well suited for, they can very rapidly take over the environment because they just clone themselves. And what better way to take over an environment than to just be able to split in half, and now there's two of you. And if you just keep adding nutrients, they just keep splitting. And then there's each of those split, and each of those split, and each of those split. And so you can, I think that the maximum amount that diatoms could split in a single day is 8 divisions. I think they've tracked some of the species are capable of something like 8 divisions in a single night. And so that's not 8 total diatoms. That's like you started with 1, and then it split into 2. So that's one split, and then that splits into 4. So that's 2 splits. 3 splits is 8. 4 splits is 16 in terms of numbers of individuals. So you can very rapidly take over a system, even from just a single cell in that case, right? So it's totally feasible that you could come in from the wind as a single cell or a couple of cells, and then become the dominant organism in a lake system pretty quickly if that's your strategy, right? So I'm going to get a picture of this Navicula because it's mostly clean from junk, and then we're going to move around and look at some of these other samples that I have. We'll see some more diverse types of diatoms, hopefully, and take a look at those. We're in focus. Just do a little composition. Very good. I'm going to slow the beam down so that the image is collecting is going to have nice, reasonably high resolution. And then I'll just take a picture. Pragmatic, hello. What's up? You're making harissa sweet potato fries and corn on the cob. Things are running low in the kitchen. Well, that sounds like okay food to me, though. I would love to have some sweet potato fries and corn on the cob. Last night, my wife made vegetarian chili for us, and that's one of my favorite things that we eat frequently. Even though I'm not vegetarian, that's how we eat chili typically. And so I'm pretty sure we're having chili again tonight because, you make a big pot of it. Do migrating waterfowl spread diatoms? Almost certainly, Boba. Almost certainly. With as flat as it is, I assume that Indiana must have had an enormous number of glaciers that deposit. Yes, the northern part of Indiana was glaciated. The southern part of Indiana is not glaciated, or not as recently glaciated, I should say. So there's like a dividing line, and most of Terre Haute, where I am, Terre Haute is basically in the glaciated section, and from here north, basically. So, diatoms can attack an environment, yeah. Elder Goth, hello. Elder Goth, are you an old goth? Or are you a strong goth, like the ancient goth. Power goth. Which is it? Asking for a friend. Welcome in. Sometimes we do light microscope streams. Sometimes I'm just watching rain happen, lightning storms, moon, sage goth. Yeah, exactly. Like a wise one. Sometimes you're an old goth. OG goth, as the kids say. Very cool. Back when goth was a thing, was its own thing. Nobody knew what to call it. Old people, yeah. Old people are cool. The young people are just trying to emulate us old cool people. With their new wave stuff. I guess. I don't know. I don't know what kids do these days, to be honest. Navicula. Perfect. I forget what I was answering. Pragmatic's question about diatoms attacking an environment, I guess. Yes, they can. Yeah, so sometimes I do some night photography streams. We look at the moon, we look at storms from a camera. I look at birds from a camera. Those are usually sort of quiet streams where I don't talk as much. I just kind of enjoy the planet we live on a little bit. I was thinking I might try to do a rain stream sometime soon. I don't know. Struggling with getting a storm to actually come through because we've just had rain. But I have some people that really like to just hang out and listen to rain and chill out. And so sometimes I do streams like that where I don't talk and I just basically let us listen to nature. Or we look at the moon. Those ones I usually do talk. We get really close-up images of the moon. Or bird streams. Those I usually don't talk, so we can listen to the birds together. Those are usually in the morning. And then pretty much everything I do is from a microscope, although we have done some macro photography streams and stuff like that. Mostly it's from the microscope. Either a light microscope or an SEM or a stereo microscope occasionally. Alright, let's zoom around. This sample was the one that didn't turn out too well. It just had a lot of clay or organic stuff over the top of it, but this one's fine. So these are from not the same material, but they have some of the same diatoms, so that'll be okay. Like this is a Gumphanema in the side view. We had this one, Truncatulum. We had it in a valve view though. This thing, there's actually two of them there. So there's one, and then a division between this one and this one. This sample was collected from a boat ramp in the Chinook Wildlife Area, also in Indiana. But it does have some Cymbella. And Studio asked me to get some images of Cymbella. These are the stigmata associated with this Cymbella. Chrysophytes, naviculoids, Cymbella, that's a pinnularia. There's lots of stuff in these. They're very diverse. I just wanted to take a look at some of these samples. These are samples that I collected on a field trip with my students, and I wanted to have some nice images for them that I could post. Oh look, that's a cool, it's like Gumphanema subtial, something like that. Looks like a flute. I want to have some nice images that I could show them from the SEM for the class. So they could see some of the little things that we actually had. The class is called Lakes and Wetlands. So it's actually about things that live in lakes and wetlands. And then we went on a field trip to lakes and wetlands. Um, and so I wanted to get some images of some diatoms from the actual lakes that we were in or around. And so these are a little bit different than the ones that we had before, but this is from a similar type of landscape. They were collected from old coal mine pits. It's just that they're from a different part of the state. Not actually that far from the other site either, so maybe Studio can also use these in her drawings if she wants. I need to rotate it about, I don't know, 30 degrees. Not bad. Oh, when I rotated it, I lost my focus a little bit though. Good. Let's do this. I want to get some brightness and contrast adjustment. It's a little bit bright. What's your question? I've heard Indianapolis was originally a swamp. Is this true? Actually, the whole northern part of Indiana was wetland, what used to be called the Kankakee. And now the Kankakee is this little tiny wetland area, basically in the north west corner of Indiana and Illinois. But there used to be a wetland that was equal to the size of the Florida Everglades that spanned from basically the western edge of Ohio all the way down to Terre Haute. And so everything across all of northern Indiana used to be a giant wetland, like the Everglades wetland. Probably bigger total footage, area footage than the Everglades were. And when people came, they didn't like the fact there was all this swampland, and they filled it in basically and turned it into farmland. So for a while it was probably very lush growing conditions, because wetlands have a lot of nutrients trapped in the soils typically. And then, then we started to add nutrients because that's how we do things as humans. But so I don't know if Indianapolis was ever actually in this sort of swamp area. I'd have to look at a map to see if, because Indianapolis is basically at the same latitude as Terre Haute, but I don't know if if you drew the actual wetland out, if it wouldn't just be on the edge of it, or whether it would actually be in the sort of wetland area. So, but to back up a little bit, you called it a swamp. Swamps are wetlands that are dominated by trees. So I don't think it was ever like a swamp. I mean, I don't imagine that it was like a mangrove type of a wetland, but I don't know. So swamp might not be the right word. Marsh would probably be the closer approximation. It was probably dominated by grasses, but that's a technical term. People usually don't focus on. Like a swamp is usually what we have in like the southern central part of the U.S., like the bayou, where you have trees, and then partial inundation, and marshes are more like grassland dominated wetlands. Uh, you watch Burbs and code your stuff. Yeah. The moon, moon and my daughter are the highlights. Okay. Yeah, she's fun to have on stream. What are the chances the astronauts left diatoms on the moon? Not living diatoms for sure, but probably they didn't. Most of the astronauts probably have some sterile. They probably were very careful about being sterile, but in terms of the clothing and stuff that they brought with them, I doubt they went stomping around in, you know, dusty environments. You're having A's and E problems. Diatoms will be 100% will be on the moon in some degree. Probably some were there. Where are you from? The Akron, Ohio area. What kind of question is that? Akib Tiff. I'm in Terre Haute, Indiana, but I did go to my undergraduate was Akron, Ohio. So, technically speaking, everything was once the floor is lava. Imagine if we humans actually consulted nature prior to choosing technologies. That would be interesting. Or if we just modeled our technologies after nature, which we do, but usually in retrospect after we've figured out how it works. Kansas has wetlands, yeah. Hi, I live very near DC, and that used to be a swamp. Yes. And DC, I used to live in DC. I used to work at the United States Geological Survey, which is in Reston, Virginia. So for as long as we're talking about where I lived, I have a history of having lived in Ohio, in Indiana, in DC, in Nebraska, and New Mexico, and Maine. But I lived in DC when I worked for the USGS for about 3 years, which is why I am a Capitals fan when it comes to hockey. So, and it did used to be a swamp as well. It's still a bit of a swamp. I don't know. The DC area in the summertime is just disgusting usually. Like the humidity is just terrible. The container of human waste on the moon? What were they doing with that? Yeah, I'm generally from the Akron area, yes. Oh, you live in Reston. Well, that's cool. The Bruins aren't really enemies with the Caps. They're like frenemies. I mean, recently they've had a lot of strife, but like the Bruins' primary rival is Montreal, isn't it? And the Caps' primary rival is really Pittsburgh. But frenemies, yeah. Reston was the weirdest place when I think of all the places I've ever lived. Reston is the weirdest because it's a designed city, and this is actually Chinook. It's a completely designed city in that like they planned it, right? A planned community, I think, is the name for Reston. It's like one of the first major planned cities in the U.S. where they planned out where everything was going to go, and then they built the city instead of our normal like sprawly approach to building things. And so if you've ever been to Reston, Virginia, you'll know like you can't find the gas stations because they're all like tucked away behind tree lines, and the grocery stores are all tucked away like in places where you would not. I mean, normally all those things are out in the main roads, and they have like an entire shopping mall, but it's like in a little pit in the center part of town, and so like you can't see it until you're basically on top of it, and that's sort of the way everything is in Reston. It's like all these little hidden things. And then there's also no place to like hang out with anybody in Reston. So like if you want to, I mean, if you're not eating dinner, you have to go someplace else to really like have any sort of community there. But I really like living in Reston because I don't like people very much. So it was perfect for me. But let's see, this is Chinook. I have one of my very close friends is also a Bruins fan, and I lived in Maine for a little while, and so I like among the hockey teams that you always have ones that you really hate, and then the ones that you kind of like when they play somebody else. I root for them. I usually root for the Bruins when they're not playing my team. So like I don't. That's why I sort of think of them as frenemies. Like I want them to do well, but like I don't know, not Brad Marchand. I wish he would stab himself in the face with his own hockey stick. But the rest of them are kind of cool, and they're like people I would kind of like to have on my team. If I was running a team, but I'm not. Let's see. This is a Mastigloia, I think. But any hockey fan is, more close to me than anybody who's not a hockey fan. So, it doesn't matter who you root for. You like hockey, I like hockey. So that immediately makes you kind of a friend. We just have different things we're rooting for. This is Acmanthidium. It's probably in the Minutissimum group. It's already at the right angle and it's in focus, so I'm just going to take a picture. I don't really care about Acmanthidium Minutissimum very much, but there it is. Let's see. Yeah. I'm writing a book about Reston. RES stands for Robert E. Simon, the founder of the town. I did not know that. Akiptif says, there seems to be a variety of different structures in diatoms. Is there an underlying reason for this? Yes. I mean, there's a reason for all structures in diatoms, and in anything biological. Do we always know what that reason is? No. Is that little Chook showing up? There's little Chook. Hi, Chook. Little Chook's one of my favorite artists, and she told me she's going to start teaching people Latin on Twitch. So if you want to learn Latin from somebody who's a great artist, that's the channel you should follow. Maybe she'll draw Latin things for us. It's good to see here, little Chook. We're going to learn Latin and art at the same time. You're going to wonder why you never learned either of them. Or never got good at either of them, maybe. So the structures that are present, some of them are because the organism, like how it lives, because the shape of the organism, I'm not trying to be glib, so the shape of the organism controls like how it lives in the environment. So some of them have radial symmetry, and they're mostly things that float in the water, and some of them have bilateral symmetry, and they mostly are things that either live attached on a substrate or crawl around on a substrate. And then some of them are asymmetric in every direction but one, and those tend to be things that basically live attached by the end that is differentiated from the rest of the structure. So the ultrastructure associated with diatoms is linked directly with the habitats that they live in and the lifestyle that they live. So if they're floating, attached, or how they attach, or what they attach to, it basically affects a lot of the sort of ultrastructure of their cell. And so your question's very broad, but that's going to be my first order answer. And then if you have refining questions, feel free to ask them. And then, we can, I can try to help explain what I mean. But it's kind of hard to answer all of that. super long name. This little nasty diatom is sort of a weed. It grows everywhere. This thing, Mastigloia, usually lives on calcareous rock surfaces, so you can see how specific things can be for diatoms. They can have highly specific niches that they live in or live attached to things on. Some of the things that are here that we've taken pictures of live on plants. Some of them live on rocks. Most of these are benthic diatoms. This is the inside view of that diatom that we took a picture of before the acanthidium. This is the Mastigloia. This is the partecta, which is the internal structure of the diatom for Mastigloia, and it actually helps you figure out what you're looking at. Somebody in the channel is that Michael-learning, I think your spirit animal was a Mastigloia. Is that true? This is Mastigloia. The name translates from Latin for those people who are super interested in figuring out what little Chook is going to learn to teach them. Translates from Latin as nipple glue. So there's your Latin for the day, nipple glue. I don't know why. Except for to say that the German people who described diatoms had some clearly suppressed fantasies about diatoms. You know, as Germans do, I guess, from the 1900s and 19th century. This is a nice internal view of this diatom, though, so I'm going to get a good picture of it, because they've got a really cool structure. This partecta structure is really kind of, it's unique to Mastigloia, and so if you see this thing, you always know that you're looking at, like, instantly. It only occurs in one genus, Mastigloia. So it's nice when we get nice clean views of it like this. Back to chat. Sorry, I have to kind of bounce back and forth because I got to run the machine. So let's see. Invisible dimensions. There's massive development in aerospace and defense industry has expanded here. 7 of the top 10 world leaders in the industry. Yeah, there's all kinds of, like AOL was there when I was there, so that was like 2000. AOL was like their huge industry that was right next door to the United States Geologic Survey. Hockey and boxing are the only 2 sports that you endeavor. That's good. Elder Goth, do you use pseudo-color to colorize the SEM images? Are you using color cues from the light microscope? I don't use any kind of... it's all colored from my imagination. So I try to use a variety of colors because some people don't like different colors. So, or I just get bored making everything blue or purple or something. So there's no reality associated with it. It's all false color. Diatoms, what you're looking at is, in nature is transparent. They're made out of opal and silica, which you can see through. It's glass basically, and so they have no color. And the color that you see in the microscope is physical color, is not real color either. It's just created by light diffraction. But so the blue colors and stuff that you see sometimes in like Pacific plankton streams, or if I do a light microscope stream, that's about how the light is interacting with the diatom. In reality, diatoms have an orange color because they have pigments, but that's not the color of the skeleton. That's the color of the chloroplast. So they're like golden brown, orangish brown, kind of like the color of my jacket. Or maybe a little bit more like a greenish version of this. And so they don't, the organism, the part that we look at in the SEM doesn't have any color. And I just add color to them to try to one, it's science art, so I do a little bit of trying to make it look appealing to people by modifying the color. And I think if I just use black and white images, I would still like it, but not everybody likes black and white images. So that's that's what I have to say about that. So it's all false color, and it's done in Photoshop and Procreate and Lightroom. just tools. So it's not based on anything. Your username is Latin. Yeah, bellum means like good, or bellum? Bellum means like pretty, little pretty white thing or something like that, I think. Be prepared. We're going to Latin and art it up. Discourse. I wish you could think of good follow-ups, but you can't. Well, we'll figure it out. I love your dog vomit emote, by the way. Michael Learning. I use it all the time. If you look in your Twitch account and you'll say it'll tell you how frequently your emotes will be used. It's going to be just me spamming that everywhere I go because it makes me laugh every time I see it. Nipple glue is the nickname for your eyes. It's good. It's not normal to talk about. I don't think it's normal to talk about nipple glue, but I don't know. You showed rectangular diatoms. What are they, and where do they grow, floating or attached? The rectangular ones were ones that were in girdle view. So that's a very good question, Elder Goth. The diatoms that we're looking at them like, sorry, I've got props. I don't know why I don't use them. This shape is a circle, right? This shape is a rectangle, but it's the same object, right? So depending on how you look at it. Hello, Disillusioned. And Minicorn's here as well. Hello, Minicorn. Ding ding, got subscriber bell going. Thank you for subscribing. But so some of the ones that we saw, we were seeing rectangles stacked like this. They're not really rectangular. They're probably boat shaped in actuality. So, but those ones that live in colonies and chains live in intermediate waters, usually between about a meter to maybe about 8 or 9 meters deep, and they're what we call tychoplankton. So they live in these sort of ribbon-shaped colonies, living attached face to face. Usually they indicate moderately deep water. When you get into really deep water, you get the sort of round ones mostly that live as solitary organisms, or round ones that live in chains that we saw, kind of like Melisira, but they have long spines that connect the faces together, which is like Oligocira. So, are diatoms anaerobic or aerobic organisms? Diatoms are algae, so they make oxygen. They also consume oxygen, but generally found where there's oxygen because they're making oxygen. That's a byproduct of photosynthesis. So, kind of like some space photos. Yeah, they modify space. This is like artsy things they do with space images. Yeah, kind of similar. Monochrome image. Thank you. You love the images. Oh, you can always get the, if you want to look at them independently, there's a Instagram for the SEM account, and I post most of those there. Or if you follow me in the Discord, there's one of the channels in my Discord. I just post pictures so you can download high quality versions of them. If you want really high quality versions of them, nobody's used it, but there's actually a channel reward where I can give you like, a 5 gigabyte picture, whatever, some massive size image you could print out as a poster if you wanted to. So, vellum means war. It has a bunch of different meanings, I think. Yeah. But you're right, I guess it does also mean war. Oh yeah, she has emotes. Yeah, good. I'm almost caught up with chat here. Disillusion's the subscriber that snuck in and got the ding dings. Very nice. I have some 3D printed diatoms. That's what this is. This is a diatom I described. This is Stephanodiscus caruscus. It's from June Lake. So I described this diatom not as a plastic piece of whatever, but as an actual diatom. Yeah, and we can print them out here, and I have a bunch. And one of my students actually made a bunch of 3D printable diatoms that you can get. They're free to download from Thingiverse, I think. If you go there and type in Joe Mohan, um, internal view, sorry, um, he's pretty good with MATLAB, so he just built a bunch of stuff, and I think mostly he has diatoms that he and I described or that I've described. And then this one I made using SEM images. I just created it by, I took about 80 photos of diatom from different angles, and then I gave those images to a guy in our department, Alex, who is a specialist in photogrammetry tools, and he turned that into a 3D model. It even has a skin and everything that matches the SEM images, but when you print it, you don't get the skin. So, yeah, pretty cool. That one is actually the organism, the way it looks, basically. It's not a model of it, other than, I guess it's, we printed one side of it, and then the other side is a mirror of it. It's a little epithemia. Actually don't recognize this species for epithemia, which is uncommon. Let's see, I need to make it like 250. 260. At the start of the stream, if you weren't here for the very start of the stream, I was actually playing a little bit of music from my friend Mel. Her Twitch name is Mel I Am, and I've been hanging out in her stream, just chatting with her. And I told her that I was really hoping at some point to use some music that wasn't pretzel rock in the back of my streams. And she said, "Oh, I'll just make you some stuff." And so she's been making me some background music to go along with my streams, just to have in the background. She does some video production, and she's a songwriter. She makes her own stuff. But we'll probably be featuring a lot of it, especially in the light microscope streams, and maybe some of the other ones. But when I start and end my streams, I'll probably do those. But I just wanted to give her a little bit of a shout out, or an actual shout out. Let's see if it works. Oops, Mel. Yeah. That one. There you go. And if you can, do me a favor and give her a follow. She's super, super fun. Her streams are very high energy, and I don't think she gets the attention she deserves. She plays ukulele and keyboards, and she's a little like all over the place sometimes, but in a very fun way. So I just wanted to give her a little attention while there were people here, and hopefully some of you will give her a follow. I have a lot of music streamer friends. I spend a lot of my time on Twitch when I'm not streaming, listening to music streams, so that's usually the people. If it's not an artist that I'm rating, it's almost always a musician, or a streamer that's relatively new. I try to focus on those things, people that I feel like need more attention on Twitch, and try to use whatever influence I can. I'm not that big of a streamer, but I can get people to check them out, and I think it's good. I think it'd be useful. I feel like if I was going to play music, I'd like to have somewhat of an audience there, to make it feel a little bit more like you can interact with people. So this species is Epithemia, or the genus is Epithemia. It's defined by the fact that it has a raphe. So the raphe is the line that they use to crawl around with. It's a slot that goes through the cell wall, and it starts up here. It's covered a little bit here, so you can't quite see the end of it, but it's this line, and it runs basically along the edge of the valve, just over the side of what we can see, until it gets towards the middle, and then basically it comes into the middle of the valve and then goes back out to the outside edge. So that's a characteristic. They sort of have this shape, and they also have these heavy costae, which you can just barely see as light areas. here, 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 here. In the light microscope, you'd actually be able to see them. But in the SEM, because we're looking at the topography or the surface of the diatom, it's a little bit harder to see through it. But you can see there's like a light band here and another light band here and another one here and one here and one here, right? Those are actually causti. In the light microscope, you'd be able to see those as thickened lines that go between them. Here you can actually see the raphe ends. So this one goes from here and rolls around to there, and this branch goes from here and rolls around to there. So characteristic shape for epithemia, basically, with the raphe that kind of comes off of the edge and into the middle of the valve. I like this diatom because it looks a little bit like a mustache, and I like putting mustaches on things in my chat. So, also, thank you, Michael Learning, for subscribing for 2 months now. Now when you chat, it should make a little bell sound the first time you come in. How has climate change affected diatoms? It's actually a really long answer to that question, Aki. There you go, see? There you go, you got the bell. It's like a little, like a personal reward you get for talking. Isn't that fun? And just the first time, but every time I start a stream, you'll be able to come in and talk and get the little rewarding bell sound. So it's actually a really complicated question, Aki, but the answer is, climate change changes temperature. It changes, in some areas it gets hotter, in some areas it gets colder, in some areas it gets much wetter, in some areas it gets much drier. So like in the place where I live, climate change makes us basically get more flooding, and sometimes we get warmer conditions, sometimes we get colder conditions, but it's the actual like flooding that actually occurs here more than anything. And then eventually it'll probably get like, the temperature of Kansas will basically work its way up into the temperature of Indiana if we keep modifying the landscape. I'm going to work on the rain stream. It's the middle of the day though, Froza. Those will happen at night here. No, this is an SEM stream. So climate change can have all kinds of influence on the lake system. What's happening with our... Oh, it just isn't updating. It's like, why isn't that updating? Climate change can have a whole bunch of influence on those lake systems. So they could become wetter, drier, warmer, colder. Most likely they'll become warmer, and in most places a little bit wetter. But in like the western part of the U.S., lakes are just going to dry up. So the answer is it's all over the place. Generally speaking, diatoms take carbon dioxide out of the air, and the warmer the conditions are, usually they do, okay, but it doesn't matter. Temperature isn't really a thriving component of most of diatom growth. They can grow in sea ice. And I think generally speaking, it probably has a somewhat positive influence on them, but like if you dry out lake environments entirely, that's not going to be positive. And it's going to create, long term, it's going to create a lot of change in the environment, which might be problematic for organisms. But diatoms will probably be able to manage to colonize the new environments because they're good at that sort of thing. So, do I want to become famous? No, I don't want to be famous. I wouldn't be interested in being famous. That's not even something that appeals to me. I just wanted people to care about science. Can you make a website that does that? Thanks for pointing that out. Sorry that haven't figured out all ways to stop the bots automatically, but it's going to happen. Diatoms already took over the world, Bellum. That happened a long time ago. They rule the oceans, they rule the waterways. You just don't know it. Diatoms are responsible for 1 in every 3 breaths you take. The oxygen that you're getting out in your body is coming from diatoms, just straight diatoms. So if we didn't have diatoms, you'd have to breathe 1/3 as much as you do. Probably it would be catastrophic for most organisms to have 30% or more less oxygen. In the water systems, when diatoms evolved, the entire oceans had to adjust to the presence of diatoms. What I mean is that silica didn't used to be something that most organisms would absorb from their environment. And so it used to be a lot more abundant in the ocean. When diatoms evolved and started using silica, they basically extracted all of the silica out of the water. And all of the other organisms that made skeletons out of silica, there weren't that many, but like radiolarians were around before diatoms. And if you look at what happened when diatoms evolved, the size of the skeletons of all of these organisms got smaller and lighter and used less silica. And they had to, because diatoms basically took over the oceans. And any aquatic environment ultimately gets dominated by diatoms. And it doesn't matter whether there's a lot of nutrients or low nutrient conditions, as long as there's silica, nitrogen, phosphorus, and sunlight, diatoms basically control that environment. People don't realize it, but they're the base of the food web for everything in aquatic systems. So fish eat them, snails eat them, all of the aquatic invertebrates eat them, everything eats diatoms. And yes, they can eat other things. Green algae are usually less appealing. They're more difficult for organisms to get the nutrients out of. And cyanobacteria usually make toxins, so they're even worse. So the presence of diatoms in the environment basically determine this sort of buffer between where the nutrients go. So if there's like a street fight and we put all the nutrients and silica in a bucket and we threw in a bunch of algae, the diatoms would take over that environment first, and they would basically suck all the silica out of the water, and then they would lose the fight to the other organisms because they couldn't actually basically compete when there's no silica. But when there's silica present, they're going to kick the butt of any other algae. And yeah, street fight, yeah, exactly. You put them all in a bucket together, cage match, diatoms are going to win that fight. They will out-replicate them, other organisms, and they will take over that environment everywhere, always. If temperatures get warm and silica starts to disappear, green algae will do better. Cyanobacteria will eventually take over because the nitrogen will get all used up, and then the cyanobacteria will basically be the only thing that can get nitrogen from the atmosphere. But in a street fight, diatoms are going to win against other algae, and they just keep replicating, right? And growing and replicating and evolving. And if we look at the total number of species on planet Earth, diatoms have an estimated something like 1 to 2 million species that is likely projected in terms of the total number. So, they have a much greater diversity. They can live in all kinds of environments. They're dominant in marine environments. They're dominant in freshwater environments, river systems. Soils have diatoms growing in them, tree bark, mosses, anything. Like literally any aquatic or subaquatic environment will have diatoms growing in them. So it's totally OP. Let's see. I'm going to scroll back up. I've been talking for a little bit there. The diatoms will be fine. Fungi will be fine. Humans, not so much. Yeah, it's our fault. And, we'll have to manage our own consequences. Have we seen dynamic and exponential change in the feeding habits or reproduction structure directly or indirectly related to our planet heating up? Yes. All of the ocean systems have changed substantially. And actually, the ocean systems in particular are sensitive to, not just the heat, but also the pH. But a lot of our major organisms in the oceans are responding to both the temperature increasing and the pH. But oceans are absorbing most of the carbon dioxide that we put in the atmosphere. It's where most of it's getting stored. And consequence of that is that we have more carbon dioxide in the oceans, which means that we have more carbonic acid forming in the oceans, which means the pH of our oceans is shifting dramatically. And that's problematic for all kinds of organisms that are filter feeders that live in the ocean. So corals, mollusks, snails, the gastropods, anything that's basically got a calcium carbonate skeleton has a more difficult time doing that when the pH changes to become more acidic. This is epithemia, sorry. Epithemia? SP, I don't know which one. Probably something close to adnata, but I'm not convinced that that's adnata. So the oceans have changed substantially, and if they weren't, we would be in trouble, because the oceans are actually without the oceans absorbing most of the heat and carbon dioxide, our surface would have already become unlivable for humans. The heat's going somewhere. A lot of it gets absorbed by our oceans. And the consequence of that is actually fairly large for our ocean systems, which were already basically overfished and have all kinds of other really significant problems that we're creating in them. But of those, probably the most catastrophic one is not the temperature, but the pH. So usually when people try to tell me, well, what's the big deal if it's a couple of degrees cooler or warmer, which maybe that's a legitimate presentation of information, because humans can move easily from one place to another. We're pretty good at adapting to our environment, and we've got air conditioning and whatever else. But most organisms don't have that power to mobilize as easily as humans do, and as consciously as we do. And additionally, we need other organisms to live. So, it might be cool that we would be okay, but we still need to eat and grow things. And fish is a major nutrient resource for basically all humans. I don't eat it, but, without it, we would have a hard time feeding people. And it's definitely a substantial problem if we create something where we collapse the ocean food web system, right? So, generally speaking, it creates problems for everything. It's not to be too dramatic, but I feel like we definitely have problems that we can identify that are going on right now associated with global warming, that scientists have been warning people about since I started studying science. So, let's see, this needs to be rotated about 40 degrees. Went a little too far. Just about right. Somewhere in here, and then I'm going to just get it into focus for us. This is a Cymbella. It's like in the Cistula group, I think. That's the stuff. Now it's in focus. Actually, it's in focus down here, but not up here. But I think that's okay, because this part's actually like the part that's exposed the most, like that elevation. It's almost 5 o'clock, and I've got stuff to do, but I want to take a few more pictures. There's some samples I haven't even had a chance to look at yet, and I think I plan on being back on maybe Friday. Friday afternoon I don't have anything planned, and normally I teach a class, but I don't have to teach it this Friday. So I was thinking I might try to get some mushrooms and get through the rest of these diatom samples on Friday. We'll see how it goes, but it's in my to-do as a possibility, so if somebody hasn't stick something else in my schedule, we might have a second stream this week from the SEM. That's what I'm saying. Okay, as pictures being collected. Sloth hair has diatoms. Yeah, yeah. This sloth hair grows all kinds of algae. That's on my to-do list for things I want. I have somebody who's collecting me sloth hair, and we will be looking at sloth hair diatoms sometime this year, I think. I'll let you know when I get the samples, but we'll also maybe be able to look at hippopotamus diatoms, because they also, I think, grow on the surfaces of hippopotami. We'll see. Hasn't been proven yet. Sloths for sure. Hippopotamus, I'm guessing, but I'm not positive. Please explain epithemia agharchii. I don't know that one, if you've read a paper on them. But epithemia, in general, are benthic diatoms. They live usually on macrophytes, and they live in places that have relatively low nutrient and especially low nitrogen. And they, as a group, have nitrogen-fixing cyanobacteria. Earlier, somebody asked me a question about, do they have diatoms that are endosymbiotic? Epithemia is the species, is the genus, the whole genus. Pretty much everything in it is has an endosymbiotic relationship with a nitrogen-fixing cyanobacteria. So, well, plankton are diatoms that are floating in the water column, and they are usually a little more successful at taking up carbon than other things. That's not totally untrue. Let's see. What are the 3 most interesting things you know about diatoms? Oh, that's a tough question. It depends on my audience. One thing I would say is, one, diatoms can, and I think we talked about this earlier in this stream, but diatoms have the ability to persevere through being dehydrated and can be brought back to life, including hundreds or potentially thousands of years, basically being in a kind of like a hibernating state and then being resurrected. And related to that is that because diatoms are dust-sized particles and they can be blown around in the atmosphere, you can actually tow a plankton net behind a plane and pass through clouds and then examine what you've collected in the plankton net from the air, and you will find all kinds of living algae, including living diatoms, in the air, among other places where you can find them. In the clouds, there are diatoms alive in the upper atmosphere, and this is partly how they get carried into new environments. I think that's pretty interesting, cloud diatoms. Another interesting thing I think we talked about also on this stream, their sexual reproduction cycle is just really crazy. And then, let's see. So they can dry out and be blown around. That's one. Their sexual reproduction cycle has this like broken wheel sort of system where it's mostly asexual and then a little sexual like link in there when conditions get right. And it controls the way their size and shape are. Let's see, what else is really interesting about diatoms? I mean, there's a lot. So I have a lot of like potential answers to that question. They're sort of fascinating in a lot of ways. I never get bored of them. I never get bored of looking at diatoms. I never get bored of looking at them in the SEM or light microscope. It's just fun. And the specimens are, again, yeah, beautiful, very detailed. Are diatoms radiophiles? I mean, they probably like listening to cool music. Is that what you're asking me? Do they like listening to cool music? I like listening to cool music. You mean like radio, do they like high radioactive environments? Oh, that's actually a really cool story. They have diatoms that they collected from cooling tanks from nuclear power plants. and a whole genus that they think evolved in the nuclear power plant water. They've taken diatoms up into outer space, and we're able to, I think, grow them in outer space for a little while. So they can, some of them can handle high doses of UV radiation, um, irradiation, but probably just for short periods. If you wanted to find something that was like really capable, you would go to lichen. There's some lichen that they've taken up into space and they've just put on the spaceship and was growing outside in outer space, getting high doses of radioactivity, and they were fine. Lichen are probably a little bit more capable of repairing that damage. But lots of things are actually, I don't know if tolerant of radioactivity versus like, those things that are actual radiophiles that like enjoy being bombarded by it. I think they're more just tolerant. So they're more like radio tolerant than something that actually kind of enjoys it. So I've ever wished that a single diatom would be as large as whales. I would like to have them that big so I could figure out what's going on with their structures, so I could figure out how they work. And also, I think they're pretty, but maybe I like them also being a little bit just like secret. Part of what I like about them is they're in this like invisible system where like you can only see them with a microscope, and they tell us so much about the actual environment. And so you need some sort of like science wizardry to actually like see them and understand what's going on. But you could hand me a sample from anywhere, and I could probably, like a freshwater sample from anywhere, and I could probably like distill what kind of environment it was without ever having known what environment it is. And I think that's actually really cool. Like, I mean, you can do that with lots of organisms, but diatoms are just really sensitive to pH and salinity and nutrients and depth and all kinds of things. And so I could just look at a sample and go, oh, this seems like it's this kind of a lake system. And just from something that you can't see with your naked eye, I could tell you all about the lake system and how it's changed through time. So it's kind of like crazy wizardry going on that I really kind of enjoy. So I think that's my other favorite part about them, is that they are so useful as ecological indicators that that I don't have to see the actual environment, and I can sort of picture what it is just from looking at the diatoms that lived in it. So it's kind of special. Look at this giant thing. Just look around for one more diatom to get a picture of, and then there's some Rhopalodia. There's a Rhopalodia. This is Epithemia now, but it used to be called Rhopalodia. Oh, it's got junk on it, and that's a Nitzia. I don't want any pictures of Nitzia. Although Studio Cornix might want a picture of the Nitzia. There's this big giant Cimbella over here. Kind of off on its own. Okay, we'll do this one. 310. I'm rotating, sorry. Gotta do some math in my head. Not quite far enough. Kapow! Now it's in focus. Okay. It's like I should have just rotated it a little bit farther. I thought we were already at 310. done fussing with the angle. I'm going to zoom in. I'm going to get us in focus again because we're just slightly out of focus. And then zoom back out. We're already composed. Perfect. Speed, I'm going to put the speed to 8 and our beam intensity down a little to 6 so you can see all of the detail in a nice high resolution image. And maybe we'll just do the brightness and contrast really quickly. This is a diatom that belongs to the genus Cimbella. This one. Everything is set. I'm just going to take the picture. So this is a 10-minute image. It's going to be a little bit longer than our normal image, and it'll give me a chance to catch up with chat because I'm sure I am behind again. Usually I am. And oh, thank you for the follow, Aki. Astronomy stream required, looking at 4 planets and the moon. I don't know if I've ever looked at all 4 planets. Are they together? Lots of critters definitely live in sloth hair. No, real size hippos. People look at turtle shells, Michael Learning. I have a friend, Shelley Wu, who sent me some material from diatoms from turtle shells. People definitely look at that, and not just Shelley, but lots of people. But she's my friend that also does that. If you can chase her down on my Twitter. She usually likes my diatom posts. She was a diatomist. Yeah, there's a bunch that are probably pretty resistant to UV radiation. Resistant to, but not necessarily like them, right? So, is there... inactive state is not called a tune state. That is just for water bears. I think they're the only organism that have that cryptobiotic state that's used that uses that terminology. Could diatoms have been sent to different planets? We could send them there, yes. I think it'd probably be better if we started off with the lichen. And if we're trying to colonize Mars, for example. Diatoms on the rain and the moon, yes. We could write a song about it if I was a songwriter, but I'm more of a song listener too. They did take hydras and tardigrades up to the International Space Station. They did that. They also took lichen and diatoms and a bunch of other organisms. Okay. Can we look at samples of diatoms to predict environmental breakdown? Yes, we can. What is paleolimnology? Paleo means past, and limnology is the study of lakes. So it's the study of past lakes. It's like paleontology from lakes, basically. And we look at fossil organisms. I use diatoms, but we also use things like ostracods, pollen, charcoal, whatever, from lake sediments to try to tell something about ancient environments. So, thick Cimbella, yeah. Okay, I'm caught up. Mostly. Launch the lichen up, yeah. We could use them to start, and then we can bring some of the other things up. I'm gonna need a bigger microscope. Maybe. Okay. I feel like we've had a great time. It's almost 5 o'clock. This image is going to take till 5 o'clock before it will be done, probably. And it's a good time for me to try to find somebody on Twitch for us to raid. So on Wednesday, what we got out there right now, I have friends playing video games. I have, which I would prefer not to raid into. I have some musician friends. So normally I would raid into Numb the Geek because he's one of my favorite musicians on Twitch. But we haven't seen Call Sign Scarecrow before, and I actually really like Call Sign as well. He's very entertaining. He's in Scotland or Ireland. I'm not sure which one. Maybe Scotland. And his name is Call Sign Scarecrow. He does all kinds of cool music, and he has lots of like animations and stuff on his screen that he made by hand. I think he used the Orin board like I do for all of my animations, and I'm excited to write into some new music streamer channel, just because I've listened to him a lot, and I sort of hang out in his channel quite a bit. And somebody gave me a gift sub because I've been there for a while, but we haven't rated him before. So I'm sure you'll like it if you like music. Samsung, hello, welcome in, and thank you for being a subscriber. You got a bell. Newt. Yes. Thanks for hanging out, everybody. It's almost a 3-hour long SEM stream. I've got a bit of a SEM headache right now because I needed to drink more. But I'm ready to go home and work on some other things. There may be a stream tonight from looking out my window at the rain and listening to some rainfall. And if there's thunder and lightning, we'll definitely do a stream. But we'll see. Okay, I let some people to thank. Michael Learning resubscribed. I got follows from Aki and Robert NST. I got some anonymous cheer that donated 1 bit. And then we got a follow from Ron LYC. So thank you people for hanging out.